As a protein biochemist, I use a lot of different software tools and databases in order to find and predict information about proteins, as well as visualize the proteins, come up with experimental ideas, and that sort of thing. Thankfully, there's a lot of free tools out there, and so here's just a quick guide to when you might want to use which. And this is a non-exhaustive list. There's a bunch of other great tools, and I'm constantly discovering more. My go-to first stop if I want to find information about a protein is UniProt. This will give you the basics, functions, links, sequence. It'll tell you about isoforms. It'll even incorporate structures so you can see different experimentally solved and predicted structures of the protein, information about interactions, and lots of links to other databases and tools. And I've done a lot more about it if you want to learn more. I've also done a lot about the PDB, the Protein Data Bank. Here you have the structures of proteins that have been experimentally solved using things like X-ray crystallography and cryo-EM. And they've now also started incorporating computationally solved, so like AlphaFold models. Speaking of AlphaFold, AlphaFold uses AI in order to predict the structure of proteins using information coming from the experimentally solved proteins and things like this. It has a database where you can find the pre-computed predicted structures of single chains of proteins. And so this is great if you have a monomeric protein, you don't have any modifications, you're not looking at interactions, you just want to see the basic predicted structure. You can go to the database and see it already predicted. If, however, you have a multimeric protein or you want to see how that protein might interact with something, then you're going to actually have to go and run AlphaFold yourself. Thankfully, it's really easy. They have a server that you can use, and it has a limited amount that you can use per day, but that'll let you use the latest version of AlphaFold. It has limited options of things that you can do, so it has like limited options for ligands that you can add, but it does have some standard ones, and it will let you do multi-chains if you have like a multimeric protein. You can also download the standalone version, but it's of an older version of AlphaFold, but you can get that from GitHub and you can even modify it. There's also something called CollabFold, which is a Python notebook-based version. It doesn't use that newest version of AlphaFold, but it will let you do more of the modifications and do custom ligands. More on that and interpreting the results in my blog as well. Now you want to actually go and visualize those structures that you've generated or that scientists have generated. There are two kind of big main tools, Chimera X and Pymol, but then there's also simpler to use kind of web-based versions that are less powerful but easy to kind of get going with if you don't want to go full in on these molecular visualization softwares. And so that would be like Molstar, which is actually what's incorporated into things like the PDB, as well as ICN3D. Chimera X is fully free for academic use. Pymol has both a free version and a paid version, but the paid version is actually, you can get it for free for an academic lab, and then the fully free version has a little bit limited of what you can actually do, but mostly you can do the same things. So Chimera X and Pymol, both of these will allow you to look at protein structures, compute distances, show things in different forms, make pretty pictures, all that great stuff. Expazy-prop-param. So here you're looking for some information about your protein, either a protein that is just like the wild type version, where you can just incorporate the UniProt ID, say, or you can paste a sequence of a custom protein. So maybe it's a protein that you've modified, a mutated version, you've added a tag or something, and you want to see the amino acid composition, you want to see the isoelectric point to plan out your purification, you want to look at the extinction coefficient to try to get the UV to protein concentration figured out. And so you can do that with Expazy-prop-param. For more information about structure, PDB-Sum is an easy way to automatically generate protein topology and domain diagrams, as well as to investigate interfaces. So you upload a PDB file or you look at a PDB file for a protein that already has a PDB file, so it's already been solved. And it'll generate a bunch of different things. So you'll get that domain architecture diagram, secondary structure diagrams, a topology map, a diagram of and information about ligand-protein and protein-protein interaction. So it'll actually tell you like this amino acid is predicted to interact with this amino acid. through a salt bridge and it's at this distance and things. It'll also predict pockets, tunnels, and channels, so PDB-Sum is a really great web-based tool. Another great web-based tool, HawkDoc. So here it'll actually go and model protein-protein interactions. And so with PDB-Sum, it'll investigate protein-protein interactions if you upload a file, a PDB file that has them. But HawkDoc, it has two kind of modes, and the first mode will actually predict those protein-protein interactions and analyze the strength of those interactions at a residue level basis. You can actually also go in and tell it, what if you mutated this, and it would then predict changes in interactions. It'll tell you kind of the interaction strengths and the contributions from things like van der Waals forces as well as ionic factors, and so that could be really great. If you have a previously solved structure or previously predicted structure, you can actually upload that, and then it'll go and it'll analyze the interactions, but not kind of do that docking step first. 3D BioNotes, so here you put in the PDB code for a structure, and this site's going to integrate all of the, well, not all of that, but a lot of the information from other databases, a lot of the kind of like annotations. So scientists figure out this does this, or this is this domain, or there's a mutation here that causes this. And then all that information is kind of mapped onto the structure in this web-based server. H++, here it's going to take a PDB file of a protein, and it's important that it actually has the hydrogens on it. And it's going to predict the protonation state of each amino acid residue at a pH of your choosing. And you can see more about it on my blog. If you're looking for information about an enzyme, ExploreEnz is the official IUBMB, so the International Union of Biochemistry and Molecular Biology. They have this database called ExploreEnz, which has information about enzymes and their classification. And again, more on my blog. Brenda also has additional information about enzymes with links to other sites. If you're looking at enzyme mechanisms, so you want to see how it's predicted to work and actually get some cool graphics and stuff, MCSA, the Mechanism and Catalytic Site Atlas, is great. Now say you want to find proteins that are similar to that main protein you care about. If you want to look for something similar in sequence, look at BLAST, so specifically blastp. It's going to search protein databases for protein sequences, taking into account both sequence identity, so are the amino acids in the sequences identical at a given location, and similarity, so are they similar biochemically? So are they both polar, are they both nonpolar, are they charged the same way, that sort of thing. You can access it directly through Uniprot. Uniprot's really great. If you want to find proteins that are similar in structure, there's this tool called Foldseek. It's going to compare structures against one another to find similar ones. And it even searches the AlphaFold Protein Structure DataBase. You can access it freely online, or you can download the code and run it in command line. And yes. Okay, so now we found those proteins that are similar, and now we want to kind of look at alignments and make some pretty figures. Clustal Omega is going to allow you to do those alignments. And if you want to visualize them more interactively, JAWView. So there's this JAWView.js, which is a web version, or a desktop version. And then if you want to make some nice figures, you can use ESPRIPT. This will incorporate like the overlying secondary structural information from PDB files. If you want to find sequences to align and get their PDB codes and structural information, then there's a kind of related software tool called NScript that you can use for that. First stop for most things with the protein, though, is going to be your Uniprot, and you can get to a lot of these other tools directly from the Uniprot entry for a protein. Hope that helped, and happy playing with protein.